All right, our last big topic in Chapter 9 is DNA replication. We've kind of hinted at this before and said a little bit about the basics, but now we'll go into it in a little more detail, and we'll describe how DNA is repaired. This first image here on the side there is a semi-conservative model of DNA replication. It shows on the top is the two old strands of DNA that are separated in the replication process, and complementary new strands are constructed from the templates provided by the old strand. So that's kind of where we're at there. Remember, DNA stands for deoxyribonucleic acid. It's a large molecule, a polymer made up of many repeating subunits. The molecule is a double helix. It has two strands. The side chains are connected by covalent bonds. The two chains are connected between hydrogen bonds of the bases. The bases pair according to the adenine pairs with thymine, forming two hydrogen bonds, and cytosine pairs with guanine, forming three cytosine bonds. Um, and this pairing of bases, this occurs in all cells, and DNA is used for the same process in all cells. It provides a stored genetic information, and that code that it stores is used to make RNA, which is an intermediate step in making proteins. And so that process is the same in all cells. It occurs with the same code using the same materials. Um, just to review, it's a two-step process called transcription and translation. Here, if you turn on the links, there's some animations there. So if you have this in the display of PowerPoint, you can get to those YouTube animations. Transcription is a single enzyme RNA polymerase that at the promoter makes contact with a part of DNA to be copied, and then using the DNA template, it it grows an RNA copy from that template until it reaches a stop sign. Ribosomes are used in the second process, translation, and translation begins when a piece of messenger RNA uh, becomes enters the cytoplasm of eukaryotic cells, and ribosomes attach to it, as does the start codon transfer RNA, which has an anticodon. And then this will use these transfer RNAs will read the codons and bring the correct amino acids to form a protein. The other really big thing DNA has to do is it has to be copied, and it has to be copied very exactly. This requires many enzymes, but the main one is called DNA polymerase. It uses a parental strand, an old strand, as a template, and it adds complementary DNA bases to make two new DNA molecules. It's a very efficient enzyme, which makes very few mistakes, but it also can repair mistakes. So it's the main enzyme, and it's the one I've introduced so far, but there are others. Okay, so here this model of DNA to the side shows nicely kind of the structure of the DNA molecule. Sugars and phosphates form the backbone. They are covalently bonded together. And what the ribosomes do is they bond a phosphate of one nucleic acid nucleotide to the sugar of another one. And these are covalent bonds. Um, and then the um, nucleotide will have an exposed base that can pair with a complementary base on the other chain. And so the two chains are going in anti-parallel directions, and the enzyme is always going to use an old chain as a template. This is referred to as semi-conservative replication. And replication happens the same way, whether it's eukaryotes or prokaryotes. It always happens like this. The strands are separated, and then nucleotides will line up by complementary bases, and the enzyme DNA polymerase will join them. This replication fork is actually, though, pretty complicated. It involves a bunch of enzymes working together. Um, Enzymes have to uh, unwind the two DNA strands. These are done by helicases. DNA polymerase is actually a large complex that will, it's a kind of a double complex that it will sit down and it will grab the DNA and it will begin working after there is first an RNA primer constructed, and that is done by RNA polymerase. So DNA polymerase needs an RNA primer, and then it starts adding complementary bases. Eventually, those RNA primers will be cut out, okay? And DNA polymerase will actually do that itself, and then uh, after it's cut out, another enzyme, DNA ligase, will heal up the strands that aren't joined. And so this is kind of what it looks like. So it's a whole bunch of enzymes working together. And DNA polymerase works by opening the two DNA strands. That's what's called the replication fork. Okay, there's a little RNA primer that's needed on each strand. One strand is synthesized continuously by DNA polymerase. The other one is done in short sections because it wants to do it in reverse. And so here you require a lot of RNA primers. These RNA primers have to eventually be cut out, okay, and replaced with DNA and then joined by an enzyme that heals nicks in the DNA. And that enzyme that joins it is called DNA ligase, okay? And these DNA ligase will join these short segments that are made in the lagging strand, the one that's not synthesized continuously. In addition, DNA polymerase, in addition to its job as linking nucleotides together that are complementary to the old strand, it can also do a little bit of proofreading. It can recognize errors that are caused by kinks or kind of expanded areas where the bases don't repair or prepare correctly. And so sometimes these are mismatched base pairs, like shown on the left. Other times that there's DNA damage caused by cross linkages between adjacent thymines. DNA polymerase can repair those. Okay, it will know that there's a bulge in the backbone of the DNA molecule, and it will back up, cut that bulge out, and allow two nucleotides to come in. And so it has a proofreading function. So it makes very, very, very few errors. Um, we have something like 3 to 4 billion base pairs, and it's estimated that when these are copied and in a new germ cell, there's really only maybe 2 to 4 errors per person. So very, very few mutations. Okay, so DNA is copied by DNA polymerase. This is just a summary. DNA polymerase works in the 5 to 3' direction. For it to get started, there has to be initiation by an RNA primer. That RNA primer is made by RNA polymerase. When the one strand is started, all it needs is one simple RNA primer, and then it works continuously um, as the replication fork opens. The DNA polymerase just keeps working towards it. On what's called the lagging strand, that's opened in the other direction, the direction opposite of what DNA polymerase wants to work. Here then, RNA polymerase needs to continually put in new primers every so long, okay, to get DNA polymerase to do these short segments called Okazaki segments, okay. Uh, these segments are between these little RNA primers that will have to be removed and replaced by DNA polymerase and then joined by DNA ligase. So there are some other enzymes involved. DNA helicase unwinds the strand. Gyrase makes sure that supercoiling is removed. RNA polymerase does the primers. DNA ligase heals up the primers after they are removed. Um, there's also other enzymes that do repairs. Now, one thing you might ask is, this is kind of the summary for us today, but you can also ask, how do these enzymes and different molecules know where to start and work on the DNA? And this is because there are a bunch of regulatory proteins that can recognize specific sequences, and they can recognize when RNA is bound to a DNA. They do this by looking at exposed bases in the major groove. So they access the DNA in this major groove, this large space, and they can actually read the base pairs and find specific sequences. Okay, so all these regular proteins have DNA binding motifs, and these binding motifs, there's several different types. You don't have to really know this, but you have to know regular proteins can find specific regions on the DNA. And these include helix turn helix uh, motifs, which have like the what's called the homeodomain, and the zinc finger motex and the leucine zipper motif. All of these types of structures allow the regulatory molecules or enzymes to find the right place on the DNA molecule and to do their job. And so it's a really important feature to understand. All right, so these enzymes were discovered not long after we discovered the structure of the DNA. Most of them were in the late 50s to the early 70s, and these form the key molecules that we use in biotechnology, in DNA technologies like genetic engineering. That's going to be our topic next week.